Hello everyone. Let's get started with our topic about genetics, transmission, risk condition or factors. The objectives of having this topic is that you will be able to understand what are the major principles of genetics, and you'll be able to discuss how transmission occur and be able to differentiate what are the risk condition or factors you're going to consider when it comes to genetics. So there are basic principles of genetics. We have what we call as the human nuclear genome. So when you say genome, genome are all of the DNA in an organism or a cell. So the size of a human genome is there are 3.4 billion base pairs, and the number of human genes, we have the 30,000, and then the genes vary in length and can cover thousands of bases. Now the average in size is we have the 3000 BP, and we have only about 2% of the human genome which contain the coding genes. So the actions of much of the genome is unknown. So we can see in the figure now the structure of the DNA. Now it's a form of spiral, and then it's helix, and we have the DNA structures. Now we have 1, 2, 3, and 4 structures. So we have what we call as the polynucleotide chain, and then we have what we call as the DNA double helix. We have the supercoding of DNA, and then we have what we call as the metaphase chromosomes. When it comes to epigenetics, this is the study of the heritable changes in phenotype. Phenotype is the appearance or the gene expression which is caused by mechanisms other than changes in the underlying DNA sequence. So these changes may remain through cell divisions. For the remainder of the cells, life and may also last for multiple generations. The chromosome facts is the number of chromosomes per somatic cell are 22 pairs plus 1 pair of sex determining chromosomes, which is equals to 46 all in all. The number of chromosomes per gamete or the egg or sperm are 23, and then 1 chromosomes of each pair donated from its parent's egg or sperm. And then we have sex chromosomes, which is the XY for males, and for females is the XX or the double X. Now the largest chromosome is the number 1, now 263 million base pairs or BP, and then the smallest chromosome, which is the Y, equals to 59 million base pairs. So we have the chromosomal morphology methods. There are two, what we call as chromosomal staining, and we have the FISH or the FESH. Now, so chromosomal banding. Now we have the human male, the G bands, and we have here the chromosomes, and we have the mitochondrial DNA, which is circular. And then we have several copies and no histones. So the function of DNA now, and then we have the gene expression. So when you hear gene expression, now that is in parallel with the term transcription or the what we call as the translation. In cells, we have the meiosis, mitosis, and the gametogenesis. Mitosis is the process by which a cell separates its duplicated genome into 2 identical halves, while meiosis, the process that transforms one diploid into 4 haploid cells. And we have the genomic imprinting. Most of the genes here express equally from both alleles. The small number of genes show the differential expression, which is dependent on parent of origin. Mainly the chromosomes number 6, 7, 11, 14, and 15. While the imprint is mediated by the methylations, which is the transcriptional inactivation. This imprint persists through cell divisions in embryo. So the embryo, imprint removed at gametogenesis, and then it will be established according to the sex of transmitting parent. The population genetics is the study of distributions of genes in populations. So you will hear when it comes to population genetics, what are the disease frequency, the genotype frequencies from the generation to generation, and then we have the genetic polymorphisms. These polymorphisms, now the occurrence of these two or more alleles at specific genetic locus in frequencies greater than can be explained by the mutations alone. While the single nucleotide polymorphism, the restrictions fragment or the length polymorphism, what we call as RFLP, and in polymorphism, the variable number tandem is repeated. Now we have the clinical genetics. This is the practice of clinical medicine with particular attentions to hereditary disorders. So, what are the inherited disorders? Now, this is caused because of what we call as the cytogenetic. Now, because of the single gene, the polygenic, and we have the multifactorial. So, in clinical genetics, you'll be familiar with the term, what we call as the genotype, which is an individual's genetic makeup, form of a particular gene at a given locus. While phenotype, this is the observable expression of a genotype. Homozygous, this is the identical form of a particular gene. While heterozygous, this is the different form of gene, which is the carrier of one normal and one abnormal. Dominant, this is a condition, phenotypically expressed in someone carrying one copy of a mutant gene. While recessive, it is dominant. While recessive, this is a condition wherein the phenotype allele expressed only in someone with two copies of the mutant gene. So, when there will be an occurrence of what we call as autosomal dominance, this is in vertical transmission. On the average of 50% of offspring of affected parent will be affected, while unaffected individuals do not transmit trait. Males and females equally affected. So as you can see in the figure. Now, what are the autosomal dominant conditions? Example of this is we have the Marfan syndrome. Okay, this Marfan syndrome is caused because of the autosomal dominance, disorder of the gene. So we have the achondroplasia, we have the familial or early onset of Alzheimer's disease, the Huntington's disease, familial hypercholesterolemia, and then we have the familial breast cancer. Now this may be caused because of these autosomal dominant conditions. While in the autosomal recessive inheritance, this is a horizontal transmission. Now, this is in siblings, but usually not in earlier generations. So, here, on the average, there are 25% reoccurrence risk. But male and females are equally affected on this autosomal recessive inheritance. Now, what are the examples of the conditions that causes of this recessive conditions? We have the sickle cell disease. Now we have the cystic fibrosis. We have the Tay-Sachs disease. We have the hemochromatosis, phenylketonuria, and then we have the thalassemias. Now these are examples of these recessive conditions. While in X-linked recessive inheritance, now these are incidence of trait which is higher in males than females. But no father to son transmission. And there is 100% of daughters of affected males are now unaffected carriers. Because of this, X-linked, now there are 50% of sons of carrier females are affected, while 50% of daughters are carriers. The trait may be transmitted through series of carrier females. So, what are the examples of this disease caused by X-linked recessive conditions? We have the hemophilia, okay? We have the Duchenne and Becker muscular dystrophy, the androgen insensitivity syndrome, the Hunter syndrome, glucose 6 phosphate dehydrogenase deficiency, and then we have the Bruton. Now, again, globulinemia. So, in XP-linked dominant, now this is different from X-linked. Now, this is XP-linked dominant. Here, there are males and females affected. Now, females usually less now severely affected than male. 1 in 2 risk to children of affected female. So all daughters of affected male affected, but no male to male transmission. Now the examples for this one, now X-linked dominant inheritance. In male and female which is affected, now we have the vitamin D resistant rickets, we have the fragile X syndrome, and then we have the OTCD. While in lethal, this is lethal in male, what we call as incontinentia pigmente, we have the Rett syndrome, the XL chondrodysplasia, and then we have the Gold syndrome. Now, in the mitochondrial inheritance, now, these are exclusively maternally inherited. Now, male and females are affected, but only female will transmit offspring. So therefore, the risk of offspring of affected or carrier females are difficult to determine, now which is 0 to 100%. Now, here, in our last discussions about these basic principles of genetics, when it comes to family medical history, now it represents that a genomic tool that can capture the interactions of genetic susceptibility, now which is shared environment and common behaviors in relation to disease risk. Okay, so these are the basic principles, now very significant for you to really know about the issue or topic about genetics. Okay, thank you.